So thank you all for being here. My name's Christopher Buck. I am the founder of Retro Report, and this is Kira Dornton, the executive producer of Retro Report, and we are the organization that produced the video you are going to see today. Retro Report was born out of a frustration in a lack of a vacuum in the space between breaking news today and follow-ups a year from now. Whenever there's a big breaking story, we will inevitably see the recap a year from now and what has happened since. Well, Retro Report is a project that seeks to create a living space for every important news story of our lifetime. And on our website, you'll see 125 plus stories now, every one of which you'll know the basic beginning of. But we complete the story. We bring the story arc to the present, and we talk about a little bit how it's affecting our lives today. So that was the idea for the project, but like any good idea, it takes somebody to actually make it happen, and that was Kira's job, and she has done just that. So the executive producer of Retro Report, Kira Dornton.
Thanks, Chris. As Chris mentioned, we're a nonprofit that's dedicated to providing the history and context that we're missing today in a lot of the news cycle. We've done 125 short docs. We partner to release them with all the major news organizations, but it's really important to us that they're released digitally, because if the future is everybody watching videos on their phone, there has to be more than cat videos available. So we like to say we refuse to surrender the digital space to subpart journalism. It's been interesting how much science and health stories have been a part. We're generalists, but science and health has been a particular topic of interest. But this series that you're about to see is different for us. It took more than a year. It's 3 segments, and we invested a lot of time and energy and resources into it. And that was largely because we really wanted to follow up on the significance of the sequencing of the human genome. And as we watched a lot of the news coverage about genomic medicine, we saw 2 forces that we're always trying to counteract, which is hype and fear. And so we put together these pieces to help the public understand what genomic medicine is, where it comes from, and hopefully what it means. It will be followed by a fascinating panel discussion. So we released it with Stat News, which is a great partner, but this is actually the first time they're all being shown together. So we hope you like them. Enjoy.
I have this very aggressive type of cancer. It's hard to treat.
Precision medicine, it's being called the future of healthcare.
Means the right dose of the right medicine for the right patient.
A super targeted approach.
It is really like a heat-seeking missile.
Expectations are sky high for precision medicine, a new strategy that tailors treatment to individual patients.
You can match a blood transfusion to a blood type. What if matching a cancer cure to our genetic code was just as standard?
But we've heard these kinds of hopes before, during the race to sequence the human genome.
The race to crack the human genetic code, which could lead to life-saving, revolutionary changes.
Medicine will be transformed.
There's at least the potential to reduce the number of cancer deaths to 0 during our lifetimes.
About 30 years ago, Marston Linehan began searching for a new way to help his patients with kidney cancer.
Now, who else in your family had kidney cancer?
There were no effective forms of therapy, no drugs that worked. I lost every one of these patients who developed advanced disease. So we wanted to identify a gene for kidney cancer, and our hope was that if we could understand the gene, that might provide the foundation for effective forms of therapy.
It was the start of the genome age, and scientists around the country were hunting for disease genes.
We had no idea what a mountain this was to climb. We set up a whole program. We analyzed DNA from over 4,000 patients. It took us nearly 10 years to find that gene.
Then a group of government scientists had a big idea. Instead of searching for genes one by one, what if they could create a map that would locate and sequence every gene in our DNA?
People are mostly hoping to see this have some payoff as far as human health.
Led by Francis Collins, the Human Genome Project was the most ambitious biological research program ever attempted. It started in 1990 with a 15-year timeline and $3 billion budget.
So the motivation really was
to enhance the ability to understand hereditary factors in all diseases as quickly as possible.
And the sorts of traits that affect the vast majority of the population, heart disease and diabetes, we know precious little about, and those are really the frontiers of genetic research.
At the beginning, we didn't have anything automated.
This is exon 5. This is a family.
Everything was done manually. You had to do all these chemical reactions one by one by one. There was radiation involved. You had to wear protection. It was horrible.
1, 2, 3, 4, 5.
The genome is made up of about 6 billion chemical letters. Collins knew without dramatically improved technology, it would be impossible to read them all.
We were, by 1998, finally where we could see this goal might be achievable. It was at that point that a separate effort was announced in the private sector.
This is the largest DNA decoding lab in the world by far.
Former National Institutes of Health researcher Craig Venter spent years trying to speed up gene discovery.
I was the first to make the transition from the old manual way. My lab had the very first automated DNA sequencers.
His announcement that his company, Celera Genomics, would sequence the genome before the government shook the scientific world.
It's going to be years, if not a decade faster.
He was using a new approach called shotgun sequencing, where the genome is broken up, sequenced, and quickly reassembled by a computer.
It's like a jigsaw puzzle with 25,000 pieces. But we knew how to put the pieces together.
Suddenly, it was a competition. Venter suggested that Collins' team sequence the mouse genome, leaving humans to him. Collins refused and said Venter's untested approach was likely to produce a genome full of gaps or mistakes.
It's not just about science, but also about the race for vast sums of money.
Collins also attacked Celera's plan to sell genome information and to patent genes, which could make it harder for medical researchers to work on them.
Somebody else who has a great idea about what that gene might do and a way in which to turn that into a product for public benefit may be discouraged from doing so.
The tension soon made headlines.
Are you going to win? We're not going to lose.
Everybody likes a good competition. It's not often that a small group takes on the whole federal government.
It actually made this effort to sequence the human genome something that people had heard about.
The greatest human science project ever.
Once it's on the front page of the New York Times that there's a competition going on, and then everybody is like, what's going on there?
Every day, it's robot versus robot, public computer versus private computer.
Both the public program and Celera were, on a frenetic pace of sequencing. It became clear that the Acrimony was only going to grow. I was starting to become concerned that this was turning into a mud wrestling match. And it was damaging both of them, and of course damaging to our program as well.
One of the Genome Project's original architects, Department of Energy scientist Aristides Petrinos, arranged months of secret meetings between Venter and Collins, hoping to make peace. And on June 26, 2000,
I am happy that today, the only race we are talking about is the human race.
They presented the genome to the world, together.
In coming years, doctors increasingly will be able to cure diseases like Alzheimer's, Parkinson's, diabetes, and cancer by attacking their genetic roots.
For researchers like Marston Linehan, hoping to use genes to find cures, it was like striking gold.
The Genome Project gave us the map. All of a sudden, we knew the sequence of every gene. We didn't have to spend years and years finding them. That was a huge advantage for us.
But it also made things more complicated. After finding one kidney cancer gene, he kept finding more and more.
We know of 16 genes that cause kidney cancer. We thought kidney cancer was a single disease. It's not a single disease. It's a number of different types of cancer that just happen to occur in the kidney.
And who is that?
Alex Din was only 34 when he got kidney cancer.
Look, this is when I first came to America. I was born in Vietnam. My parents always wanted to give me a better life. The week that I was diagnosed, we just found out that we were expecting our third kid. So it was, this week of roller coaster emotion. I had my left kidney removed, and then it started to spread to the other side of my abdomen. So then we said surgery is not an option anymore.
Alex and his wife are doctors. Wondering why he got cancer so young, they went for genetic testing.
They tested the entire genome and found that I had this one specific mutation. Rare gene, kidney cancer is rare, I get them both.
Through a friend, Alex heard about Dr. Linehan, whose lab was using genes to develop treatments for kidney cancer.
Once you discover a cancer gene, we take tumors from patients and we study how the cancer gene works. And then we work on the development of drugs.
In this case, drugs for the type of tumor Alex has.
And when you go here after treatment, it's all dead.
And if they look good, then we go to clinical trials in patients.
How are you doing today?
Good, thanks.
4 years ago, Alex joined one of those trials for his specific cancer, one of a small but growing number of cancers that can be treated with this type of targeted drug.
I'm still able to take care of my family. I'm still able to work. I mean, without it, I know I wouldn't be here still. So.
It's all you might call personalized medicine. It's targeting that specific gene mutation instead of just trying this drug and that drug and this drug.
But critics, like epidemiologist Nigel Paneth, point out few patients are likely to be as fortunate as Alex.
We haven't yet found very many examples of drug interventions that depend upon genes. There are 1 or 2 here at the... So for the last 60 or 70 years, we've added about 10 or 11 years of life expectancy in the US. None of that is due to genetic discovery.
That's because, after years of research, it turns out the relationship between genes and most serious health conditions is more complicated than expected.
Heart disease, schizophrenia, some of the common cancers. There was a hope that while those would not be the result of a single gene, they'd be influenced by a short list of genes. Maybe for diabetes, there would be, 4 or 5 genes. We're up to 82. Each one of which has very
What role genes play in most common diseases.
Francis Collins of NIH, here.
As head of the National Institutes of Health, he's helped start a massive new federal effort called the Precision Medicine Initiative.
So the time's right to unleash a new wave of advances in this area, in precision medicine.
It will employ dramatic advances in sequencing.
The year Dr. Collins helped sequence the first human genome, it cost about $100 million, and today it costs less than $2,000.
And supercomputers to sift through the genomes and health records of about 1 million people, searching for links between genes, the environment, and our health.
Their genes and their environment, and they somehow come together in a way that causes somebody to become ill or not. So if we really wanted to get a complete understanding of what causes diabetes or the common cancers or Alzheimer's disease or mental illness, you've got to look at both of those things.
But Collins and the government still have competition.
We're processing, on average, a complete human genome every 15 minutes. And we set a target goal of trying to get 1 million genomes by 2020.
Craig Venter now runs Human Longevity, genomes and health data, hoping to pick up where the original genome sequence left off.
It's just not a direct linear line of the genome was sequenced, we found this gene, and now X. And what we're discovering now are the things that occur maybe once in the human population out of 10,000 or 20,000. It's the rare combinations of things that determine our human traits.
But it's hard to know how much impact those discoveries might have.
Huge amounts of data don't guarantee that you will find the things that matter. In fact, they increase the chances that you will find tiny things that don't matter very much. It won't be a solution to diabetes, hypertension, heart disease, lung cancer, breast cancer, because those are not largely determined by the genetic inheritance we get.
For those who've been part of the genomic revolution,
without these medicine, I'm sure I wouldn't be alive today.
There's no question but to push forward.
Well, it's like we're coming out of the dark ages. Medicine's been based on rough symptomology and a little bit of hocus pocus without knowing the true molecular nature of what's going on.
My name is Carl Zimmer. I'm a columnist for the New York Times and a book author. Those are my dulcet tones you heard in the background on the show. And we have a really wonderful panel here of scientists who are dealing with the issues that the code talks about and explores in detail. And so since we had some technical trouble, I'm going to sort of introduce each of them and in the process bring up some of the key themes of this series. Let me just, I'll start with Della. Della Makower is a medical oncologist and clinical researcher at Montefiore Einstein Center for Cancer Care, where she specializes in treating patients with breast cancer. She's an assistant professor of medicine at Albert Einstein College of Medicine, and she recently served as the co-chair of a study called TAILOR-X, which is a study on trying to figure out the best kind of medical care for particular kinds of breast cancer. And this kind of gets at what we were watching earlier about how cancer is increasingly going towards a molecular level of analysis. So maybe, Della, maybe you could tell us briefly about the TAILOR-X project. Well, actually, let me back up and ask you, because this is a very historical project, and I'm sort of curious for you what the contrast is like compared to when you started in treating cancer. Back then, how much was there, trying to figure out what particular kind of cancer a patient had versus now?
Well, so I think that there's been a lot of changes from when I started taking care of patients with breast cancer until now. And as far as figuring out what kind of cancer a patient had, there are a few sort of classic prognostic markers that we look for in breast cancer: hormone receptor status, HER2 status, tumor grade, which is basically how aggressive a tumor looks under the microscope, and then also we think about the anatomic stage, how far the cancer has progressed in the body. That was sort of all we had when I first started taking care of patients. And there were randomized studies looking at adding chemotherapy to patients with early breast cancer that showed that chemotherapy added some benefit as far as curing patients with early breast cancer and eradicating what's called micrometastatic disease. But we knew that, especially for women who have the hormone receptor positive subtype of breast cancer, we were overtreating a significant proportion of women. But what we didn't have was the tools back then to actually be able to tell someone up front if the chemotherapy would benefit them.
So it was just chemotherapy for everybody?
So it was pretty much chemotherapy for everyone who agreed to it based on stage of disease and their general health and their age. So patients would say to me, is this going to work for me? And how do you know if this is going to help me? And I would have to say to them, there's really no way to tell. All I can tell you is the results of the studies that we have, that if we take a large number of women with your stage of cancer and we give half of them chemotherapy and we don't give the other half chemotherapy, we cure more women who have gotten the chemotherapy. And then the next real advance was the advent of multi-parameter gene testing, and what those are, they're tests that look at gene expression in tumor tissue.
So that means which genes are turned on?
Yeah, basically, which genes are turned on, which of them are transcribing RNA and making proteins, and there are a variety of these tests that are commercially available. And they can subdivide cancers, and we can learn more about the biologic behavior of that particular breast cancer by looking at those tests. And they generate a type of a signature that can be used to tell us information about prognosis and can also be used to tell us information about benefit from chemotherapy. And one of these multi-parameter gene tests is a test called the Oncotype, which looks at 21 genes, 16 cancer-related, 5 reference genes, and measures their expression by RT-PCR.
Which is what?
Reverse transcriptase polymerase chain reaction. Okay. So basically what I do as a clinician is ask the patient, tell the patient about the test, ask if they would be interested in having this test run, and then send a piece of their tumor to a lab that runs the test on, and it comes back with a recurrence score, which is a number that goes from 0 to 100 and is both prognostic and also predictive of chemotherapy benefit. So for patients who have a low recurrence score, what we know is they have a very good prognosis. If I give a woman with a low recurrence score chemotherapy, I don't make her good prognosis better. I just give her side effects. Then there are women with high recurrence scores. Those women have a more aggressive type of cancer and they have a poorer prognosis, but those are the women who are really impacted positively by chemotherapy. Those are the women whose lives are saved by chemotherapy. And there were a group in the middle that really constituted the majority of patients with hormone receptor positive early breast cancer that we didn't really know what to do with. And so, and what I would get back from when I would talk to my patients would be a piece of paper with their score on it, with the graph, and with where they fit in on the graph, and where, how patients like those would have fared in, sort of retrospective studies when looking at tumor specimens when they got chemotherapy. So the TAILOR-X study basically was a randomized trial of over 10,000 women, where women with early node-negative, hormone receptor positive, HER2-negative breast cancer agreed to have this test done on them. They got the recurrence score back, and then they were asked to, if they wanted to continue on with the study. Women who had a low recurrence score of 0 to 10 automatically did not get chemotherapy. Women who had a higher recurrence score of 26 and up automatically did get chemotherapy. And then the women who were in between were randomized to get chemotherapy or not chemotherapy. And all the women got surgery and hormonal therapy and the other standard of care treatments. And those women were followed for several years. And earlier this month, the results for the randomized group were presented and published. And they showed that the majority of women who had a intermediate range recurrence score did not benefit from chemotherapy.
So if they got this particular score, you could just say you can get them hormonal therapy.
No chemo.
No chemo. And how many women are we talking about maybe who could benefit from this?
So so there are about 260,000 cases of breast cancer. About half of them are node negative and hormone receptor positive, and the women who are in the intermediate range are about 2/3 of the women with breast cancer. So that is a lot of patients.
Right. Having had this experience now, do you think that there's going to be more opportunity for cancer treatment to become more personalized? I'm not sure that's the right word, but at the very least, like taking advantage of this molecular information to say to individuals like, you get this therapy, you're not going to get this is a waste of time?
I think for this particular type of cancer, yes.
Okay. Very cagey.
Question?
Yes. Let me. No, absolutely no. But I want to introduce you, so I'm going to scroll down. So this is Vinay Prasad. He's a hematologist and oncologist. He's assistant professor of medicine at the School of Medicine. Oregon Health and Sciences University. Vinay is also, I would say, a master of the tweet storm. So if you're on Twitter, you need to follow him because he is.
I apologize already if you.
No, he's a force of nature on Twitter, addressing a lot of actually these issues, believe it or not. So, so please, just
I guess,
jump in.
And I'm curious how you feel about this, but I guess the way I would put Taylor RX, which I think is a very important practice-changing landmark study, is that before we had it, there were a number of factors we used to advise women about whether or not they should get chemo, like their age, size of the tumor, hormone status, comorbidities, et cetera. Now we have one additional piece of information, which is the status, but the question is still a population average question. You can't tell a single person you will benefit, you won't benefit. You'll just say, in the old days, we know on average women with ER, PR negative tumors that are more than 3 centimeters, they tend to benefit. Now you say, we know that those people also with oncotype 20 and higher, 30 or higher, they tend to benefit. But it's still the same sort of thing on average. For the individual, I think we still have that gap. Would you agree, or?
Well, yes and no. I think nothing, there is nothing more important than an individual discussion between a woman and her doctor. Be that as it may, this is, I think, it's a huge practice-changing study. I think that this is another big, sort of piece in the puzzle I think it's, I'm not quite as, I guess, nihilistic about it.
I wouldn't say nihilistic. I think it's another important information, but fundamentally the task is the same. Using all the pieces of information we have, you tell a person, on average, people like this do this. But you still can't say, for you, this would happen. Because we just know, for instance...
Well, we can't predict the future,
exactly.
Yes,
and also, like, even within Taylor RX, if a woman is younger than 50, those patients tended to benefit. So you can't say just absolutely based on this.
Right. So for that subgroup, yes, for women who are younger than 50 with an intermediate recurrence score, those women did have some benefit from chemotherapy. But I think it's now a fundamentally different discussion than it was before Taylor RX. Because before Taylor RX, it was, well, we don't know what the benefit is for you with this oncotype. Now we have a number. It's a small number on average, and it's much more of a discussion of, are you willing to go through the side effects of chemotherapy for this small percentage benefit?
Let me jump in. I want to bring in Richard Gibbs to my far left. So Dr. Gibbs is a professor at Baylor College School of Medicine. He's the director of the Human Genome Sequencing Center. And Dr. Gibbs was actually quite involved with some of the stuff we saw on the video with the Human Genome Project, and is now using the human genome for clinical testing. And so I'm sort of curious for you, like, it's about 20 years since you were working on the project, and now you're using it to help patients. Was that faster than you thought? Was it slower? I mean, when you look back at the history, what's it like for you now?
Well, of course, some parts faster and some parts slower. And you heard Francis's great quote about technology's been, overhyped, underperforming the short term, overperforming the long term. I think we're seeing that happening. The areas that have gone spectacularly are mainly in pediatrics and with relatively simple genetic problems. The areas we're still struggling with are with cancer and with adult disease like heart disease and lung disease. So in one area we're doing extremely well, in the other area we're catching up. Well, I do want to say, I think we've got to step back. I see these films and I get a bit depressed because I hear people say, we're not making progress or it's not really genetic. I think we shouldn't lose track of the fact, the overarching fact that we are genetic creatures. We are programmed by our DNA. DNA will help us to understand and fix these disease problems. It's a hard thing to do to understand all the nuts and bolts, but we will get there. So in pediatrics, I think if we look at that, we really have got there. A decade or so ago, if you had a child, 1% of children have some sort of genetic problem. Maybe 10% of those, or 5% perhaps, had an avenue that could bring them to resolution of what their problem was and bring them on a journey to intervention of some kind. We're up to 25 to 50% now. We've revolutionised the methods, we've trebled the catalogue of genetic diseases, and we're using that information in a compound way to understand adult biology. So that's good news.
So what's like a typical case that you have now where you're using genome sequencing to treat a child?
Let me tell you about my grandniece. So my sister called me 6 years ago and said her child's second child was not doing well. And we just then developed the methods where you can sequence the parents and the child and pick up new changes in the child. And the short story is, we did that for my extended family. We found a particular mutation in a gene we thought was suspicious. We went to our database, we found 3 other cases, and we linked the families together. We never would have done it just by the clinical picture alone. But with the molecular data plus the clinical picture, we said, aha, we've got it. It's a new syndrome.
So what's involved? I mean, is it something with their metabolism, or what?
So we know the gene is involved in transcription and DNA repair. We're on a journey now to understand it and bring about treatments. But now we have 100 families worldwide who have this disorder. So we think there might be about 200,000.
So this is in effect a disease that didn't have a name. I mean, you find there are symptoms that tell you that something's amiss, and you find a gene with a mutation. It sounds like you really don't know how that mutation causes the disease in a detailed way yet. Yeah. But you're finding other people with this disease that no one knew existed before.
This is probably the most important thing I can say today, if people get this. That is a critical moment. Once you find this fundamental defect, you then have a platform to build all of your understanding, all of your treatment approaches, basically everything you want to do if you're ever going to get to a cure for that disease. Before you find this fundamental thing that's wrong, you got no idea.
And how do you go from this one child, your grandniece, to all these other people who lo and behold have the same condition?
Well, first up, we extend genetic diagnostics into the community so it's available. I said we have 100 cases. They all come from the US or from Germany, from Holland. They're not. We haven't got the cases from China and India yet. So we extend the technology so it's accessible and available. Then we aggregate the cases. We make molecular models, cellular models, and animal models with the disease, and we study its nuts and bolts so we really know what the fundamental machinery of the defect. We have a change in a letter of DNA, and we have delayed speech. How do you link those? You have to do it through this chain of investigation and discovery.
And is there any sort of treatment yet that comes out of finding that mutation?
Well, in this case? I think just as you heard this beautiful story about your breast cancer trial, as we do this fundamental discovery, we have the more empirical approaches that are testing different variations on therapy. And so for sure, we're able to reach out now to the library of drugs that are already available to affect other conditions, and in the laboratory ask, is this gene now touched by any of those drugs? Could we ever bring in a drug that's already approved to work for these children? We're doing CRISPR in the laboratory. Can we cure the cell, and then can we cure the mouse, and then maybe one day the human? So all of these things come to bear. So
potentially by editing that faulty gene, that might be a way of treating the disease in the future.
That's the dream. You know, whether it's fixing the gene or whether it's a drug because the gene is doing its damage too early, right, or whether it's a drug that substitutes for the function or inhibits the function, that's the open question.
So I want to bring in Robert Green. Robert is a professor of medicine at Harvard Medical School. He's the director of the Genomes to People research program. The other day you were talking about how you want to do genome sequencing not in the case of a sick child, but a healthy child or a healthy adult. You are seeing this as a much broader kind of application of genome sequencing. So why would that be a good idea?
Well, everything Richard said is absolutely correct. When we have these sick children and we can build a bridge to the molecular cause of their illness and then work your way through the chain of events that create the disability, that's where you're going to discover some very specific treatments. But there's a whole nother avenue of this, which is illnesses that have not been identified as genetic because they don't fit the schema of a monogenic sort of dominant or classic recessive or classic X-linked disease. So we started off sequencing healthy adults, and we found a remarkable 20% or so of them, that's 20% of the people sitting in this room, are carrying a mutation in a disease-associated gene. And these are healthy people. They're walking around just like you without any named illness. But then when we went back to those people and we said, you know, this is a funny gene that it's supposed to affect eyesight. Have you ever had any problem with your eyesight? They go, oh yeah, you know, I can't see at night. Or we'd go back and find an abnormal blood test that had to do with their iron transportation, or a dozen other odd things. So they had subclinical presentations in many cases of these mutations. There's just a lot more genetic disease in this room than you can possibly imagine. And we've been doing the same thing with now newborn healthy babies, which, by the way, is really hard to get through IRBs. And through the FDA. But when you do that, we found another 14% of these healthy babies are carrying the same thing. And of course, in this case, some of these diseases haven't even declared themselves in any way, shape, or form. But even in the babies, we could circle back and find some subclinical presentations of the disease. So this makes me think that you know, there's often been this dream that everybody's going to get sequenced. It's going to get really cheap. Everybody's going to do it. And half the world says, oh my God, that's a horrible notion. And the other half of the world, mostly the West Coast half of the world, says, well, of course that's going to happen. It's a great thing. And why hasn't it happened already? And I live on the East Coast, but I guess I lean toward the left coast on this, because I do think that we're going to learn a lot that informs prevention of disease as well as treatment of conditions that we already have.
But if you're finding subclinical stuff, is there a need to deal with it medically?
Oh yeah, because for example, I did one study on Framingham individuals, because that's like a time travel. You know, the Framingham. The Framingham Heart Study is this famous study that's gone on for 50 years of this community group in Framingham. It's developed much of the knowledge we know about heart disease. And they took DNA on a lot of these folks, and then they've painstakingly followed them over the course of their life, second generation, now third generation. So you can look at their DNA and then look back across all the diseases that they've gotten. And we found people, for example, who were carrying a mutation for cardiomyopathy, a thickening of the heart wall. And we could see on echocardiograms their heart walls getting thicker and thicker and thicker over time. You know how that usually declares itself? Sudden cardiac death. So you can't echo everybody. Nobody's going to line up an echo machine right here and do an echocardiogram. But you could identify the two people in this room who don't know they're carrying a mutation for cardiomyopathy, and you could echocardiogram those two.
So what do the rest of you think about going all in for genome sequencing, healthy as well as sick?
Should we have a show of hands?
Yeah, let's do it. Who's all in favor?
Wait, for the audience or two?
Sure. All in favor of all healthy people getting their genome sequenced.
That's great, because I have a phlebotomist outside.
Who's again it? Vinay. And I'd say, I'm guessing, a tenth or less of you. Okay.
I mean, I'm not against it in the sense that I don't think it's worth pursuing in the context of research, which is what it is, research. But before, I think, as a society, we choose to invest in such a thing, there has to be at some level demonstration that it improves outcomes for people and doesn't do the things that you worry about, which is unnecessary anxiety, stress, downstream procedures, monitoring, for instance, your echocardiogram, but not changing the outcome of sudden cardiac death. God forbid, that's a possibility. You know, so I think that those of us who've been in medicine, I think, a long time have seen many things down the pipeline over 10, 15, 20, 30 years that we all thought were highly promising, that when you studied it rigorously, it didn't do what you thought.
And that is an excellent point. But think about it. If you take your genome, you are amortizing the value of that over your entire lifetime. So you're a brand new baby, you're looking for pediatric diseases and pharmacogenomics over your whole lifetime. You get to your reproductive years, you want to use your recessive carrier traits to avoid having a child with a terrible disease. You get to your middle years, you're trying to avoid cancer. You get to your later years, you're trying to anticipate and enroll in clinical trials or even treat, if we have treatments, Parkinson's and Alzheimer's disease. How do you do a collection of, let's say, a randomized trial that is supposed to last 70 years?
I guess I would say, that's probably not the first trial I would start with. I mean, I would start with the very simple trial, which is something that we are facing right now, which is that of all the patients with solid cancers that fail standard therapies, there are companies that sell sequencing of those tumors. And the idea is that a fraction of those cancer patients will be paired with drugs that may benefit them.
So I should just to jump in. So these companies are looking for these particular mutations that have arisen in these tumors, not just in the rest of the person's body, and those tumor mutations may make it them better targets for certain kinds of medicine.
Correct.
And that's the idea.
And that's the idea. And clearly, some of those mutations in certain tumor types do that, do confer sensitivity, but whether or not they work the same in different types of cancer is unknown. But we are facing that question as a society, should we pay for this? Which is a very small step down this path, I think.
Yeah, but really apples and oranges. You're talking about treatments of tumor. I'm talking about essentially apparently healthy individuals.
Della and then Richard.
I think the other thing we would have to decide as a society before we went down that path is what is going to happen sort of socially. Can these people get health insurance coverage? Who will pay for annual echoes in these patients, in these people? They're not patients yet. Can they get life insurance? Those are the can of worms that you open when you start doing sequencing of healthy people. And I think before we go down that full force, you have to answer. We have to think as a society of how we're going to deal with those issues.
Richard?
Well, I think as a matter of position and conjecture, you can take a strong position to say we should do this, and you can deal with physician paternalism and with the economic obstacles. But we may be spared all that just by the inevitability of this. As we lower the costs and the difficulty of the test, and we improve its usefulness in a very small way in many, many arenas, it's very likely that we'll all have our genome sequenced. Maybe we have a small skin spot that we just have some concern about it. That sequence gets into our data, or maybe we're having children, our carrier test gets into the database. And pretty soon we're all going to have our genome sequenced. We won't be giving them out to the public, which is the question we might have added to your question of who wants their genome sequenced, who wants their genome sequenced and put it on the internet, who wants their genome sequenced if it's in their medical record and potentially of use for them, and it only costs $100. I mean, that's the extreme question.
Yeah.
So I think we're going to get to that second point and obviate much of this important discussion.
Right, because I guess now, traditionally, if you were concerned about something, well, as a clinical geneticist, if you were concerned about a particular symptom, you're like, that might be such and such, and then you would order a test for that.
And you could order a much more limited test for that.
Yeah, for maybe one gene or just a few genes, and now you might as well just sequence the whole genome while you're at it.
The basic thing is we're moving from a world in which genetics was rare and expensive to a world of genetic abundance, where this will be available to so many companies and so many people. At least two of the people in this room have put their genome on the internet. One sitting over there, Esther, as part of the Personal Genome Project of George Church. And the sky hasn't fallen in. There has been all these privacy concerns, all these discrimination concerns, but and we should not be cavalier about them, but they have not thus far, knock on wood, caused widespread difficulty.
Yeah, I put my genome online as well, and
you did.
I'm okay.
All right.
So,
it's three of us.
So far.
But are you healthier? You haven't benefited, have you?
I have intellectually benefited. I got part of a book out of it, but
oh well, that's tremendous ROI.
Wait a minute, Vinay. How many people
professional benefit? But no, it has not helped me. But maybe. Let me.
How many people in this room benefited from newborn heel stick? Possibly nobody. There's one.
Okay.
But that's,
That's
to
where
ask
you're
each
looking for
individual person.
a particular set of these hereditary diseases like PKU and other
Yeah,
things.
but to ask each individual person, did they benefit, is really unfair.
But you just asked each individual person, were they harmed, which is equally unfair. So, okay, so it's the same thing.
You ready to switch back to the film?
We have solved the technical difficulties.
All right.
Go down and come back?
Are we going to come back or are we done? We're done. Okay. Thank you so much.
So we left off, if you guys remember, it was the Precision Medicine Initiative. So there are 2 additional parts. The next part is on gene editing, which is gene therapy and CRISPR. And then the third part is on genetic testing.
This is a milestone that could one day erase hereditary conditions.
Just like you can change a single letter in a Word document, now we can change single letters in the DNA.
It's called CRISPR.
Clustered, regularly interspaced, short palindromic repeats.
The new gene editing platform, CRISPR, is generating a lot of excitement.
CRISPR could help rid us of disease.
But this isn't the first time scientists have tried to fix the genes that make us sick. It started over 30 years ago.
Gene therapy.
The medicine's new frontier.
It was the first time they were going to be altering a human's DNA.
The expectations were crazy.
I like reading as well as playing chess, and I like to go to different places. I would want to make people more aware about sickle cell.
How are you, Pierce? Pierston Charles has sickle cell anemia. It's a painful genetic disease that distorts his red blood cells so they can't deliver oxygen to his organs.
His first blood transfusion was 7 months old. He had a stroke. He lost his spleen, and shortly after the gallbladder, he's had a total of 48 hospitalizations.
He's one of about 100,000 people in the United States with sickle cell. Most are African American and on average live to their mid-40s.
There's no definite treatment for sickle cell.
Sickle cell was one of the first hereditary diseases to have its gene mutation or sequence change identified.
Ever since I met my first patient with sickle cell disease, that's been really my challenge to say, let's find a cure that is based on the cause. We've known the sequence change since the early 1970s, but I'll point out that's over 40 years ago, and we don't have any therapy based on knowing that sequence change.
So in his lab at Stanford University, Matthew Porteous is trying to repair the gene that causes sickle cell.
So what we have to do is edit some blood stem cells.
He's using a new gene editing technology called CRISPR-Cas9.
Ready to electroporate.
It works like genetic scissors, cutting out and replacing a gene's misspelled bits of DNA.
We can change single letters in the DNA and convert the disease-causing variant to a non-disease-causing variant. The cells that have been edited are these bright green cells.
Pierston needs monthly treatments to replace his sickle-shaped red blood cells with normal cells. He donates the old blood for Porteous' research, which he hopes could someday help him.
They're attempting to modify the genes so they can cure sickle cell. I think that it's pretty amazing.
Porteous has applied for approval for a clinical trial.
We think these cells represent cures for patients. They do everything that they're supposed to do. So that gives us confidence that it's worth trying in a human patient.
We have our fingers crossed. We're excited.
But along with CRISPR's promise come fears about its accuracy.
A DNA break could be dangerous. It could cause other mutations.
And its potential use editing genes not just in patients, but in embryos, which would impact future generations.
We're talking about something that would affect human evolution.
Critics worry CRISPR could be used to create designer babies.
You may want to make a baby taller, faster, smarter.
The power of this new CRISPR technology has resurfaced questions that people have been thinking about for 40 years. If we had a tool that would allow you to change the DNA that would then be passed along to next generations. Is that something we should or shouldn't do?
Many of the questions and the controversy remind Ashanti Da Silva of her own experience with a different kind of genetic engineering called gene therapy nearly 30 years ago.
Ashanti, come baby.
When I was born, the umbilical cord was infected and I would turn blue a lot.
She was born with ADA deficiency, a rare genetic disorder that crippled her immune system. Although there was no cure, her doctor had an idea, though it could take a lifetime.
He said, you know, there's this potential trial going on at the National Institutes of Health. Let me send her blood.
And he told me, Mrs. Da Silva, this is for possible gene therapy when your daughter is 60 years old. And I thought, oh, how wonderful.
Unlike CRISPR, which repairs genes, gene therapy would send in an extra healthy gene to do the work of the defective one. But before Ashanti could be treated, there were scientific and ethical hurdles to overcome.
They had told my parents there'd be risk of leukemia, death. It was the first time they were going to be altering a human's DNA. We all went to the NIH, and they actually didn't have FDA approval yet. It was really up until that day. They were waiting for the phone call. I was in the room.
This is on the 14th of September. A great day for the world, a great day for medicine. Gene therapy has been approved. You can read that.
Our daughter is the first patient in the world to receive gene therapy.
They had taken my cells and injected the vector with the corrected gene into my cells. They transferred those cells back to me to see if they would give me a new immune system.
How about that? Okay.
My parents did receive death threats because there was this idea that altering someone's DNA is playing God.
There was a historic medical first today. Genetic engineering as a potential way to cure an illness in a human being.
The gene therapy revolution seemed to come almost instantly, fueled in part by discoveries from the Human Genome Project, the national effort to find and sequence all human genes.
Payoff has already begun by the discovery of a long list of genes.
As researchers found more and more of the genes that cause hereditary diseases,
it is only a step toward eventual treatment, but it is a significant one.
Gene in a cancer patient.
News stories touted gene therapy as the potential cure.
Gene therapy to improve a potentially deadly condition,
Such as muscular dystrophy, sickle cell anemia,
and perhaps even AIDS.
We presumed as a society that if we had the gene, we'd be able to then quickly move to a treatment. The expectations were crazy.
James Wilson was one of gene therapy's pioneers.
In these human cells, he was able to replace the defective cystic fibrosis gene with a normal gene.
In 1999, at the University of Pennsylvania, he began testing a gene therapy for a rare liver disease that mostly affects boys.
On their first meal, they develop a coma, and there's 50% mortality as a result of that.
To deliver the healthy gene into the patient's body, Wilson used a specially prepared virus called a vector.
A young man by the name of Jesse Gelsinger received the vector. Things went terribly wrong. He mounted this enormous immune response that led to his death. I had spent almost 10 years trying to make it safe. That's probably irrelevant. What is relevant is the young man died.
After hundreds of gene therapy experiments, Gelsinger's death came as an enormous shock.
Every researcher in America was reminded this is messing with the code of life.
Why aren't they reporting these?
Congress held hearings, and federal agencies investigated.
The FDA says Penn researchers failed to report previous toxic reactions in humans and the death of test animals in similar experiments.
I wasn't given all the information, and some of the information I was given was not true.
Penn's program was shut down, and Wilson banned from clinical trials for 5 years. Then investigators began uncovering problems at other institutions around the country.
Several others died. Hundreds more had side effects, but researchers didn't tell federal health officials.
Gene therapy also faced scientific setbacks.
A cystic fibrosis treatment, a muscular dystrophy treatment, a brain cancer treatment all failed.
To this day, gene therapy has yet to cure anyone,
including Ashanti De Silva. The benefits from her gene therapy diminished with time, so she still needed regular enzyme injections to support her immune system.
Gene therapy, at least initially, did give me that boost that I needed, but it wasn't what they could call a cure.
The tragic death of Jesse Gelsinger was one of several events that led to a precipitous decline in the support for this field. Support just evaporated after that.
It seemed that gene therapy's prospects were over.
Here's an area of the liver.
But a small group of researchers continued on.
Our first responsibility to everyone, including Jesse Gelsinger, was to try to figure out what happened and why.
Can you put on our low magnification?
We learned that the problem was the immune response, not to the gene, but to the delivery vehicle itself.
Wilson returned to the lab, hoping to make safer vectors.
And the vectors that we generated seemed not only really safe, but were much more efficient. I knew that they would be a game changer, and we just distributed these to my colleagues. And I hoped that there would get traction. It didn't take long to discover them. It took a lot longer for there to get traction.
An experimental gene therapy is...
Almost 20 years later, gene therapy has been experiencing a revival and is showing promise for conditions ranging from hemophilia to Parkinson's disease.
One more.
Peter McConnell has a genetic eye disease called choroideremia.
It leads to tunnel vision, in some cases blindness.
He's the father of 2 young children, and hoping to see them grow up, joined a clinical trial at the University of Pennsylvania.
When I initially was approved, I didn't think twice about it. I was just going to do it. They take a genetically mutated virus and they inject it in the back of your retina. 4th letter, maybe a V. The goal isn't to miraculously restore eyesight. What they would consider successful was stopping the eye from progressing and getting worse. I'd be more than happy if that was the case.
For the first time ever, the FDA has just approved a gene therapy treatment.
The first gene therapy was recently approved in the United States for a form of childhood leukemia. But because gene therapy doesn't actually fix broken genes, no one knows how long its benefits will last.
What we're asking to do here is to cure diseases, and whether we can ultimately achieve a permanent genetic graft that reverses their disease forever, we'll see.
That hope for a permanent cure is why so many are excited about CRISPR.
Wow, human genetic engineering. Yesterday was science fiction, and today it's reality.
Get this, it could eliminate chemotherapy. It's fast, cheap, and can cut and paste genetic code with great precision.
All the results say that it is working. The ultimate test is going to be those first few patients. They're the ones taking all the risks.
Patients who take risks, like Ashanti De Silva did with gene therapy. She's grateful for the treatment she got, but regrets gene therapy's long hiatus.
When you're doing something like altering someone's, DNA, there are risks, but it gave me life. I was able to complete my master's, get married, have a nice home life, so I've been very lucky.
As for James Wilson, he is now working with CRISPR, but remains mindful of the lessons from gene therapy's early days.
That period of exuberance early on in the development of a new, novel, cutting-edge technology, it's very easy to get caught up into that. The reality is this is still science, and these are still experiments, and we are still learning.
And now we're going to have part 3 on genetic testing.
The genetic testing industry is booming.
The more you know about your DNA,
the more you know about yourself.
There is a simple genetic test that could show you whether you're likely to get a serious disease.
DNA tests are now being sold for everything from lactose intolerance,
farewell dairy,
to cancer.
It's like a cancer stethoscope.
But how well do they predict the future?
I could get stomach cancer. There was an 83% chance of it.
I definitely thought you have the gene or you don't have the gene. I did not think there was any gray area.
excited for class today? Yep, I can feel the energy. There were 5 kids in my family. My brother Steve is the one who had the mutation and passed away.
Massachusetts teacher Beth Lambert lost her brother in 2006. He had stomach cancer caused by a rare mutation in a gene called CDH1.
The next step was for all of us to be tested. There are 4 remaining children in my family. 3 of us tested positive for the mutation, and we knew our choice was to have our stomachs removed. If I didn't do this, I could get stomach cancer. There was an 83% chance of it. How was your day, Ben? I was 38 years old, married. I have 2 kids. It was not a difficult decision because we watched our brother suffer.
For Beth and her family, the promise of genetic testing is clear.
1% of stomach cancers are of this type. At that point, I think only about 100 families in the world even had it. There are so many people who have a genetic condition and there's no solution. I feel so grateful that we have a choice.
Life-saving genetic tests were among the things scientists hoped for when they sequenced the human genome in the 1990s.
It'll give you power over your own life instead of just randomly waiting for symptoms to appear.
By the time the Human Genome Project was unveiled in June of 2000, it seemed as if testing would soon be available for almost any ailment.
We are going to know why I'm at risk for one thing and you're at risk for another.
Silicon Valley pioneer Ann Wojcicki is a big believer in the power of genetic testing to help keep us well.
What I want to know is what's the best way for me to live? Like, what am I at risk for? How should I modify what I'm doing so I can be as healthy as possible?
In 2006, with backing from Google, she started 23andMe, selling direct-to-consumer DNA test kits.
I believe that the consumer is capable enough to get this information on their own. Everyone could be a geneticist. Everyone can understand it. This is what you get in the mail. It's super easy. You get the little tube. You spit in here. We tell people what the variations are in their genes. The first product was 12 reports. How much Neanderthal do you have? Which parts of the world do you come from? Prostate cancer risk, Alzheimer's.
23andMe saved my life.
While 23andMe sold directly to consumers,
The Neogen company.
the use of DNA tests by doctors was also growing.
Breast cancer runs in my family.
BRCA1 and 2 screening for breast and ovarian cancer is one of genetic testing's best-known success stories.
The actress Angelina Jolie went public about her choice in an...
To avoid cancer, women who test positive can choose extra monitoring or to have their breasts or ovaries removed.
As for Angelina Jolie, her risk of breast cancer fell from 87% to less than 5%. Kelly Donahue was diagnosed with breast cancer when she was 37 years old.
I had the most wonderful life you could possibly have. And this kind of put my world to a stop. Did you give the boxes? They sent me for our genetic testing. If you test positive for either BRCA1 or BRCA2, the recommendation is to have a double mastectomy, as well as a removal of your ovaries. I do remember getting the phone call and saying, oh, what's the news? Is it positive or it's negative? Well, it's not positive, Kelly, and it's not negative. You have a variant of unknown significance. And so now, what does that mean?
We've been collecting data on the BRCA1 and 2 genes, and there are hundreds of variants in those genes that we don't understand today, that no one understands.
Geneticist Heidi Reen runs ClinGen, a data project that analyzes test results and symptoms from thousands of people, hoping to figure out what gene variants or mutations mean.
When we sequence an individual, they have about 5 million variants. That means 5 million spots where they're different than someone else. And the big challenge is trying to figure out which ones are actually going to cause disease and which ones have no clinical impact. If you're wrong, that can have huge consequences. There are patients that may have had their breasts removed unnecessarily. There are patients that could have had a device implanted in their heart. And that patient may never have been at risk.
Kelly's gene test was inconclusive, but because she did have breast cancer, her doctor agreed to a double mastectomy.
But now, I had to worry about my ovaries. I didn't want to hear 5 years later, oh, you know that variant that you have? Well, now it's positive, and then, already have cancer in my ovaries. In the back of my mind, I had this worry. So I just kept going to appointment after appointment until finally they said, okay, we will take your ovaries out.
Hi. Hi. Good to see you.
Harvard geneticist Robert Green studies how genetic tests are perceived and used. He says Kelly's decision reflects long-held beliefs.
Thinking about having multiple children, we went and had ourselves genetic tested.
The early low-hanging fruit were diseases where if you had a mutation, you definitely got the disease.
Tay-Sachs testing will be held in your community soon.
And this gave us a narrative in genetics that we could do this for many, many types of diseases. But it's just much more complicated than we thought.
Okay.
Recently, Green has been testing healthy people, and he's found that sometimes they can carry a well-known mutation that we expect to be dangerous without getting sick.
You have this mutation in, for the disease chondrodysplasia punctata, and yet you have had no complaints that would be related to this condition.
I'm almost 60. There's no symptoms, so no, I don't worry about it.
He had a mutation which is associated with a bone disease. Now, he doesn't have a bone disease.
For doctors and patients, it's hard to know what to do with these kinds of results.
What happens if you have a mutation for a cardiomyopathy, a thickened heart wall, but you've never had a symptom in your life? Should I be concerned? It's a completely unknown question.
Green cautions that testing's ability to predict future health may be most limited when it comes to the conditions we most worry about.
For the vast majority of heart disease, diabetes, and almost all of the common cancers, there simply isn't one gene or one gene change that's going to predict whether or not you get that.
Ironically, at the same time scientists were discovering limits to their predictions, the business of DNA testing was booming.
32 companies advertise on the internet offering to test your genome for a host of diseases.
Tests that promise to predict your future, your risk for cancer, for other diseases.
By 2013, 23andMe had tested over 500,000 people for an increasingly wide range of ailments.
Users can find out about more than 250 health conditions and traits. The results cover everything from lactose intolerance to Alzheimer's disease.
But the Food and Drug Administration determined that 23andMe's results, along with results from other direct-to-consumer tests, were not clinically validated.
In terms of what the results mean, for many of the claims being made by these companies, the science is not yet sufficiently evolved.
The FDA then sent a scathing warning letter ordering 23andMe to halt the sales.
23andMe had to stop selling health tests, and several of their competitors closed. But to save her company, Wojcicki worked with the FDA. Out of over 250 reports, the agency has approved just 13 health risk reports and a few dozen for carriers of disease genes.
We have had to change everything. We also have a limited subset of what we used to have for reports, but we are working with the FDA to try to get more reports back. They want to make sure that the data is accurate and then make sure that people understand it.
The FDA is developing new regulations for consumer medical tests. But in the meantime,
your personal DNA nutrition test.
Testing your genome is not just about diseases.
Genome is revolutionizing the pursuit of wine through genetic testing that pairs personal taste to perfect bottles.
And it's big business. Companies make money selling tests,
unprecedented insights about your fitness potential,
and also selling users' DNA data to drug companies for research.
Part of a market that's expected to generate $600 billion globally by 2020.
23andMe, whose millions of users fill in elaborate health surveys, has several lucrative partnerships.
Could be Parkinson's with one company, it's lupus with Pfizer. The combination of data from the surveys and the data from the genome will enable us to do massive, huge studies. I do think that there's a race about data and genetics and really understanding what you do with that, and everyone's running fast.
A or a G.
Whether it's for medicine or commerce, nearly 20 years after the first genome was sequenced, scientists are still trying to understand what our genes mean and how to use them to improve our health.
Hello again.
Now we're sequencing millions of genomes and learning something new with each successive one. Over time, we will understand what this information means. Our hope is that we can identify true risk for disease at a level where an intervention makes sense.
The way it did for Beth Lambert.
We were thrilled to have that information and to be able to do something about it. My brother didn't have a choice at all, and it's the greatest gift he could have given us. The surgery literally saved my life.
Let's plug them in. So if our panelists don't mind, we'd love to go back up on stage for a few minutes. We have about 10 minutes left for Q&A. So if the panelists will join us up on stage, we're going to have a handheld mic.
Thank you.
I'll just put in a plug. If you want to hear even more about genomics, I'm giving a talk tomorrow at noon. So please come to that if you're interested.
Yeah, we're going to go straight to Q&A. Can we have microphone ready? Here. Microphone here. All right, let's start here.
Is there a way to use DNA testing in nutrition, that is to match your DNA with your diet?
Can you match your DNA to your diet? Who's going to take it?
The short answer is there are lots of DNA tests that will reflect on scientific aspects of diet, but there are companies selling this with the implication that it actually helps you modify your diet, and that is probably an overstatement of value at this moment in time.
Anybody else?
No, I'll leave that alone, I think.
All right, this side. How about this gentleman right here?
I actually just want to make a comment. I'm a genome scientist, also a good friend of Richard's. I think one of the things that has not been discussed, healthy genomes are unhealthy people who have disease. That the way this is going to make a big difference is for the data to be available, and that means both the clinical data and the genomic data and the readout of the genome, not just what you were born with, but as Dell talked about, RNA and the expression of the genome. And while that's getting better, I think practically anything from NIH now, like the All of Us project, these are data that, and it doesn't mean companies only, it means everybody. So much of this is still going on in academia. And 35 years ago when I first got into the field, you could not get data from clinical studies, from anything genomic. People guarded it, and we learned from the Genome Project that sharing this is it's not letting 1,000 flowers bloom, it's letting 1,000,000 flowers bloom. It's a huge, huge issue here, and I hope everybody who cares about this will help advocate for that.
Yeah, well, all of us, Robert, explain to us what all of us is, and then Richard.
So he referred to the study All of Us. President Obama's Precision Medicine Initiative has two components, the Cancer Moonshot and the All of Us research program, which is 1 million people who will volunteer to be followed like a giant Framingham study, including their whole genome sequence.
Yeah, and to follow on, so what we don't know in the near future is whether the All of Us program and the other discussions and implementations of access to clinical records and clinical genomics will be the major transformative force, or whether some of these more direct tests and the open public sharing of data will make a huge contribution. So that's something we've all got to wait and see. But you'd be surprised at how much personal information and genomic information are being uploaded to very public websites by individuals right now.
Vinay or Della, do you want? Okay. Woman right here has her hands up.
Thank you for the panel. Could you comment on how epigenetics is interfacing with some of the things that you said, and what were the implications for environmental modifications as well as gene modifications?
So we've talked about genetics. How about epigenetics?
I think the cancer people should say something here, because we all would we could give you an hour lecture, each of us, about epigenetics. It's important, and it's a component of our kind of inheritance. But some of the most exciting stuff is happening in cancer. you want me to tell you what's happening?
Let's get a cancer person.
Well, we're all interested in looking at markers for somatic change. What happens between your germline and in cancer? And sometimes it's DNA changes, it's somatic mutations of the changes of the letters, but sometimes it's changes in parts of the genome that just express themselves differently or shut down. And those things are epigenetic changes in the somatic lineage. They mark cancer, they cause cancer, and perhaps they're pathways to treatment in many examples. So very exciting. Having said that, epigenetics is often just a surrogate for we don't know, so it must be something else. Epigenetics is really exciting. So you're not going to be able to take an epigenetic pill to cure something too soon, I'm afraid.
The only thing I'll add is, I mean, the reason I think we were both a little reluctant is to take the question is that, you know, to my knowledge, the only real epigenetic treatment that we use for patients is something called a histone deacetylase inhibitor, which is a fancy way of basically saying if every gene in your genome is a light switch on the wall, this pretty much walks in and just flips a bunch of them. And we don't know what it's flipping, but it has some modest effect in certain cancers. And that's really kind of clinically, treatment-wise, as far as, you know, I know of its role.
Can you imagine a better targeted kind of epigenetic treatment where you take these drugs and you basically say, okay, go switch that one on, turn that one off? I mean, or is that just fanciful thinking?
No, it's there in the cell, maybe. Some of the second-generation CRISPR-related technologies are activating individual promoters and switching on individual genes. So the notion that we can, you know, control genes with not every light switch, just the ones we want, is actually there in model systems. Getting it into advanced therapies is a much bigger journey.
Okay. Another question? The woman right here had her hand up.
Hi. With the advances in genetic testing, it's become easier and easier and easier to diagnose prenatally things like my son's condition, which is Down syndrome. The problem for me is that while this progress is admirable and so on, it feels like a march towards ridding the world of people like my son, who is a perfectly wonderful young man who has written a book and has a job and has a life and lives independently in a house. And you can see him in the film they're running tonight at 7:30. And so what's the balance between improving our capabilities of doing this and still having an awareness of who we want to eliminate and who we don't, and why? It's a very, very difficult and thorny question.
And this is not a new question. I mean, this is the basis of eugenics for over a century. So
yeah, but I mean, when I hear statistics like over 90% of prenatal diagnoses are being terminated just because of the diagnosis without the concomitant research, meeting other people, finding out what it's really like. There are still so many old wives' tales and so many stereotypes going on that it worries me, because with the advances in early intervention and education and that sort of thing, people like my son have an increasing ability to live a normal life and be included in the community. So are we trying to get rid of these people, or are we not?
Who would like to respond?
Well, of course, you're asking a huge question that we're not going to solve in this discussion, but I would make two very quick points. One is genetic literacy is key to understanding the nuances of all of these issues, and I think most people would not put Downs in the same category as some of the very, very painful, devastating, very early deaths that children have due to genetic disorders, and the same methods are useful for all of them. But I think this is why we have meetings like this, to discuss these very tough other issues.
I'll just add, I mean, I think you're asking a very deep and profound question that's been asked for many years now. I think those of us who do not have disability think that being in a disabled state has often very low quality of life, which study after study shows that that is not the case. And many people like your son are living very rich and full lives. So I do think we stigmatize disability, and I do think it runs the exact risk you articulate. There was a philosopher of science, Adrienne Ash, who herself was blind, who for many years, spoke passionately about this issue. And so I do think that's a very legitimate concern, and I thank you for really raising it here.
So we're going to have to wrap it up. Okay, so thank you to Retro Report for this wonderful series, and thanks to our panelists, and thank you for coming.